first thing that we do is we take a confluent T25 of ES cells. So I'm just going to wash with PBS. And I'm just going to add a few drops of trypsin. Let that sit for a couple minutes. And I can see that the cells are coming off the plate, so it's pretty much ready. Okay. So now I'm going to collect the trypsinized cells, and it's a good idea to triturate your trypsinized cells for just a moment to break up the colonies. And put them onto the ungelatinized plate. And this will just sit in the incubator for about 30 to 45 minutes. Now I'm just going to spin down these cells to collect them. And I'm just going to aspirate the media. So I'm just going to resuspend in a few mils so I can count the cells.